Okay, thanks to the organizers for giving me the opportunity to speak. I will be presenting what we are starting with LentiCure. LentiCure is a company, spin-off company from Erasmus MC for developing gene therapy for Pompe disease. And I will start with these high prices of gene therapies that you can see nowadays, not only well, for various diseases, and you can see here 4.25 million, 2.1 million. This is per patient. This is going to be a problem. We have now on the left upper side you see len-meldy, as it is called here, or lip-meldy in Europe, for a disease that's very close to Pompe disease, metachromatic leukodystrophy, or MLD, and yeah, that is really worrying. There's also for spinal muscular atrophy, another muscle disease, AAV gene therapy, extremely expensive. And why is that the case? Well, this depends on the business model that is used to develop these gene therapies by the industry. And normally investors are investing their money with the goal to get a return of their investment, and that return should be several fold of what they put in money-wise. And yeah, gene therapies are not like enzyme replacement therapy. They work for a longer period, or even lifelong, if it is as you wish. And if you talk about a rare disease, to obtain a large return of investment for relatively few patients, you need to increase the price to get that return of investment. And that is the system, how it works. But yeah, the consequences are the high prices. And what are the consequences of the high prices? That we are actually at a starting point of gene therapy for many genetic diseases. We have now technology with different tools, actually, technological tools that are now available to develop gene therapies for really a lot of genetic diseases, not only Pompe. And if we go on like this, you can feel it coming that the system can implode. We cannot go on with asking prices of $3 million US or euro per patient, or in that order of magnitude. Insurance companies will stop reimbursing the therapies, and then we have a problem. You have to be very rich, or you don't get the therapy. But actually, another consequence is that the regulatory authorities are delaying approval because they don't like the high prices, and they ask for additional analysis, but that actually further increases the cost and will delay the availability of the therapy. And companies are now actually withdrawing the gene therapy field and focus on therapies that are commercially more attractive because of all these hurdles. One example is Orchard, who has actually withdrawn from 2 therapies now. And the final result of this is that gene therapy, while technically possible, will not become available for patients or for all patients. And this is now, we are actually on the edge of what's happening in the field, and well, we try to do something about it with LentiCure. So LentiCure is actually aiming for a double innovation. First, to develop lentiviral gene therapy for Pompe disease, and we are also working on Hunter disease, which is another lysosomal storage disorder, where we are actually at the same level right now. And that is a new development, so that is one innovation. It's not there yet. And on the right you can see that we are aiming for a new business model, an alternative business model that we are setting up, like a social enterprise. We are not aiming for return of investment. There's actually no investment. There are stakeholders. That's what we're looking for. So this will be a social enterprise, and the social enterprise is not only going to develop this gene therapy for rare diseases, but also finding out what are actually the costs, because nobody knows what actually the costs are. So we will make kind of a blueprint for developing gene therapy from an in-house spin-off company. So here you see it. That's our aim, to bring gene therapy to patients against cost price and a socially acceptable margin. So we are not interested in profit. We have founded LentiCure. It is 100% owned by Erasmus MC. Erasmus MC is, the board of directors is strongly supporting this and is also realizing that we need to do something to change the system. And so we are depending on different kinds of funding, foundations, government, insurance companies, angel investors, etc. And we go for social pricing with transparent pricing and reasonable pricing. And actually, in our statutes, we included this as our major goal for LentiCure. And it looks like this. We have a CEO, Dirk van Asseldonk. He's from the business side, so it's actually he's leading the business unit of LentiCure and doing all the work that I don't want to do as an academic. I will push the science forward from the lab. We have Ans with Hannerieke and Nadine, with very strong clinical team where we can do the clinical trial in-house. We have many patients and many clinical data. We have Isabel on the right. She's doing the regulatory affairs, doing all the license work. And on the left we have Bodil Willemsen, and she is CMC expert, so responsible for making the lentiviral gene therapy product and with all the specifications. And she has 10 years, worked for 10 years at Novartis. So from the other side, and now turning to LentiCure to help us. And she worked on Kymriah, the first lentiviral gene therapy product on the market. So she knows what this is. And we are collaborating with the Leiden University Medical Center with Professor Staal and Lancaster. How much time do I still have, Nina? A few minutes?
A couple of minutes.
Good. Science. Here's the science. So, it doesn't look very scientific, but this is the cartoon of how it works. So what are we talking about? Left up, you see the patient. So from the patient, we are taking a bone marrow cell. So this is a classical bone marrow transplantation. In the bone marrow, you have hematopoietic stem cells. So these are bone marrow stem cells who can live lifelong. Then we take them out the patient, we culture them for a very short period in the lab, and then we put the gene, in this case the GEA gene. We do that using a lentivirus, and the lentivirus has the property to be able to insert the gene into the DNA of the stem cells of the bone marrow. So that's what's happening. So these are the green circles that you can see. And the end is that you have your hematopoietic stem cells that are now corrected. They express the GEA gene. And now we transplant the cells back in the patient. The cells are derived from the patient itself, so therefore are not rejected. And now the patient is actually producing the enzyme in its own body. We have seen that also with AAV gene therapy. Then you use the liver, and here we use the hematopoietic stem cells. And also then it's a form of enzyme replacement therapy, but then continuously produced by your own body. So the stem cells will secrete the protein into the blood, and from the blood it goes to the target tissues in a continuous way. Now there are two advantages of this therapy. The first is that it can also cure the brain, and this of course is extremely important for the classic infantile patients. Normally there's a blood-brain barrier. You can see it here on the left. That's a cartoon of a blood vessel, and there are cells around it, and the cells actually seal it off for large molecules. On the right, shown with a wall. But an enzyme replacement therapy cannot cross that. The protein is too big, so the door is closed. But the door is actually transiently opened by the procedure used for the bone marrow transplantation. So in this procedure, you temporarily weaken the blood-brain barrier because that's the preconditioning method that you need to do to get the therapy working. And that actually weakens the blood-brain barrier, so the cells from the bone marrow can pass over the blood-brain barrier and can differentiate to brain-like cells, like supporting cells, microglia-like cells. And they will stay in the brain, and they will secrete the missing protein and cross-correct the brain cells. And that has been shown also for the disease I just mentioned, MLD, that's strongly affecting the brain. And even indeed in patients this works. It's not only a lab finding. So we are very enthusiastic about this because it's a big advantage compared to other therapies. And we actually made an improvement to the lentiviral gene therapy project by fusing the GA gene with an IGF-2 peptide, and these are the white bars. And with the white bars, you can see here like a dose response in the heart on the left, in the muscle in the middle, and in the brain on the right. And it's like a dose response from right to left, and you are looking at glycogen levels. So you can see when we don't put the modification, you see the glycogen levels. You can remove them to a certain extent from the heart, the muscle, not actually from the brain. For some reason, it's not enough. But when we use this fusion protein with the IGF-2 tag, we can completely remove glycogen, and there's an exceptionally well high efficacy in the brain. We can completely remove it now. You can also see it here. If you look at the right upper figure, that's the brain. And the black dots are the untagged version. And on the x-axis, you can see the number of viral copies in the bone marrow. And on the y-axis, glycogen. So whatever we do, we don't really get rid of the glycogen. And one dot is one mouse. If you look at the white squares, this is what the IGF-2 tag. So you can see that we can, at a very low number of integrated copies, we can completely remove the glycogen. And on the bottom you can see a muscle biopsy with that shows also correction. The second advantage of this therapy is immune tolerance induction. And we have shown that and published. You can read it in the publication below. So we all know that antibodies can form against the GA protein. And this is actually measuring the antibody. So on the left you can see the detection of antibodies if you start enzyme therapy in mice in this case. Then if we do first a gene therapy and then we after treat the mice with enzyme therapy, then the mice have become tolerant. They don't make antibodies anymore. So why is that? Because they get kind of a reset. Because of the bone marrow transplantation, if you perform a bone marrow transplantation and you give them the protein of interest, the GA protein, then the body recognizes that as being self, and then there are no antibodies formed. So after that you are actually tolerant to antibody formation. So that's a huge advantage of this therapy. And this is my last slide. So the circled people have played a very important role in the preclinical data that are shown. So Chusi Liang, Eva Vlaar and Fabio Catalano on the upper right, or the guys on top, is the new generation continuing here. And we have funding. We are part of the Center for Lysosomal and Metabolic Diseases in Rotterdam. Thank you.